teaching back in Bangladesh for around 5 years in this particular area of topic. So I tried my best to give you some more detailed information within that stipulated time. I think you definitely will enjoy the lecture. So the contents, I was dividing it into two parts. So in the first part, we definitely will be discussing some introduction, and we will be also discussing about the staple foods, that is our area of interest, and the agents that are causing that food loss and food security issues. We will be discussing detailed about this. And we will also be discussing few things about insects, although it's not my area of interest, but for this lecture purpose, I'll be giving very few information about that insect. And definitely for plant pathology, we will be focusing in the first part the basics of plant pathology. And there are some very important issues you should know on that particular topics. And we got very few quiz. And definitely there will be a break for 5 minutes. And after that one, it will be vast details of some of the major threats for security in terms of qualitative loss. And we will be discussing about the mycotoxins, which is a threat for the quality. And how can we diagnose the disease, we will be discussing a little bit. And in between, there should be a few quiz. And at the end, we will be discussing about the management issues and some of the challenges that we need to address. So there is a link in between the last module and this module. So last module was talking all about the food security. I think you must know about the 4 pillars of the food security. So my area of interest on that food production and the food loss due to that pest and pathogens. So before start that lecture, we should know the world population and the rise of the world population. And if you see the graphs, then you can easily figure out the population increase. Definitely it's more in the developing countries than in the developed countries. But by 2025 to 2050, there would be around 9 billion people around the world. So we should feed all these people, and that feed should be sufficient and should be safe food. So just a small recap of staple food and staple crops. So the staple food actually, the majority of our daily intake from that staple food, and 60% of this food usually come from that cereal crops. Cereal crops including that rice, wheat, maize. And there are some tuber crops like cassava and potato. And few other things, fruits and vegetables are also included here, and also some animal foods. We will not be going details of that. So what are the annual loss? In the statistics, although it's kind of varied, but in between 35 to 40% food loss occurred due to some pest and disease. Pests include insects, weeds, and definitely there are some post-harvest losses. So our main focus would be on the disease loss. It's worth around $220 billion. Imagine how big loss it is. And these are the major agents that are causing that food loss in terms of quality and quantity. So the major groups includes pest. So if you see on your left side, the definition of the pest, it includes insects, mites, nematodes, rodents, slugs, snails, as well as birds. And plant pathogens like virus, bacteria, fungus, and some molecules, they are also included in that plant pathogens group. And also the weeds. So in total, combinedly they are called pest. So if we separate pest into insect as a pest, then the definition of the insect is that they are basically belongs to the arthropods group. Arthropods means that jointed legs. And the insects are hexapods, means it has 6 legs. And the insects are both beneficial and harmful. So our target of discussion here is the harmful part of the insect. And in the parasite, there are 2 kinds of parasite, plant parasite and animal parasite. So what is parasite? Any organism which is depend on another organism for their food and their nourishment and also for their habitat. So these are parasites. So either that should be plant parasite or animal parasite. And if you divide these 2 groups, then you can find that either they would be obligate parasite or facultative parasite. So what is obligate parasite? If one parasite completely depend on one particular organism, then you can say this obligate parasite. And there should be some facultative parasite which will be part of their life cycle would be completed on a particular organism. So we'll be discussing further in our next lectures, next couple of lectures. And our today's main topic would be the plant disease. That plant disease caused by plant pathogens. And if pathogen attacks on the plant, then there should be a disease. So what is disease? The abnormality of the host normal growth and development is called plant disease. So in total, if we divide total crop loss by 2 factors, one is biotic factors and another is abiotic factors. So we will not be discussing about the abiotic factors. Our target would be to discuss the biotic factors. And in total, there would be a stress caused by that biotic factors is called biotic stress. So this is a very good article that mentioned about plant pest, which is considered as the biggest threat for the food security, and also in other name it's called biological threat. So in this article they were mentioning about insect and pathogens and disease. So to give you a very brief glimpse of insect, you should know the life cycle of the insect. So the life cycle is divided into 2 categories. One is complete metamorphosis, another is incomplete metamorphosis. In the complete metamorphosis, if you see that it started from the eggs, then larva, then pupa, then adult. So in this category, the larval part and in some cases the adult part is the most destructive part. Not the eggs, not the pupa. And the larval stage, sometimes it's called caterpillar, sometimes called borer. Okay? So, and it takes around 1 to 2 months to complete their life cycle. And in the incomplete metamorphosis, it started from the insect, then nymph, then adult. So in this group, both nymph and adult, they are harmful. If I give you an example, then you all know about the locust, plant locust. These and grasshoppers, they are belong to that incomplete metamorphosis group. So in that article, these insects are considered as biggest threat for the food security. So the desert locust, this group of pests are considered as the worst pest for the history. Why? Because in previous 2-3 decades, these pests actually causing enormous crop loss, particularly in the sub-Saharan African areas. And you know, in the last lecture, the Phil Salisbury, he was discussing many of these insects, so I will not be going details. And I will mention about another important pest is shoot and fruit borer of the eggplants. So I also put the question mark. That question mark actually indicating that is that all of the pest? No. There are many more pests in the world, and it depends on country to country, continent to continent. So these are not the all worst pests. There are many more pests involved. So our today's area of discussion start from this, particularly from this point. I mentioned two very good review articles. If you get time, then go through of these two articles that highlighting the importance of plant disease for the food security, and there is a very good link with the climate change and food security. So these are very highly cited review articles. If you read through, then you could better understand about the importance of plant disease and plant pathology. So you should know some of the brief history of plant disease, because it was long before it was causing a very good loss of the crop. And in our religious book, in Bible, you know that it mentioned a lot of diseases about the blast and blight, but at that time they didn't know the cause of the disease. So, and a great philosopher, Democritus, he also mentioned about plant blight, and father of botany, Theophrastus, you all know, he mentioned plant disease as the curse from the god or punishment from the god. And in Romans, as they suffered a lot from that plant disease, that's why they created a separate god, his name is Robigas, to worship prayer so that God will not give further punishment in terms of plant disease. In between there are some other history. I will not go further details, but definitely I should mention about the father of plant pathology. It's Anton de Barry. He was invented that popular Irish famine caused by late blight of potato, and he was considered as a fungus. But at later stage, it's that fungal group separate from other group we will be discussing also. But anyway, he proved that a particular pathogen is responsible for a particular disease. So that was a very big invention at that time. And Leeuwenhoek, you all know about, he is father of bacteriology. Why? Because of invention of the microscope. So without microscope, we could not see bacterial structure. So with the invention of the microscopes, there was a big revolution for the microbiology science. And Jacob, he was mentioned as father of nematology. Nematodes are also biggest threat for our food security. So start from the plant pathogens. We discussed about what is pathogen. And if you just take a look of the plant cell, you can imagine the size of the different pathogens. What are the pathogens? They are virus, they are bacteria, they are nematode, they are protozoa, and also molecules. If you see that the viral groups, they are very tiny compared to the big host cell. So without ultra microscope, like electronic microscope, you never see the structure of virus. And bacteria, you need a high power microscope to see the bacterial structure. And for the fungus, you can see it's comparatively bigger than the bacteria, and on top it's nematode. So nematodes are the biggest plant pathogens. So you can just get an idea of the plant pathogens. So I will be mentioning here about 3 categories of plant pathogens. One is necrotroph, one is biotroph, another is hemibiotroph. I will just write these terms. So these 3 categories are the major categories of plant pathogens. So necrotrophs, they are taking nutrients from the dead materials. The biotrophs, the pathogen requires living host. That is why it's called biotroph. And hemibiotroph, they are in between biotrophic and necrotrophic. Both characters remain in that hemibiotroph. Okay, so then there are 3 very relevant terminologies. One is pathogenicity, pathogenesis, and virulence. What is pathogenicity? It's the ability of the pathogen to cause the disease. And in pathogenesis, it's chain of events of host pathogen interaction to cause the disease. So we will be discussing about the life cycle of the disease that will be belong to that pathogenesis groups. It's a few more steps involved to cause the disease. And in the virulence, it's the aggressiveness of the pathogen to cause the disease. Not all the pathogens are aggressive to cause a disease. So there are some virulent, there are some avirulent pathogens. Okay? So we should know about the plant pathogens and their multiplication. So if I start from fungi on top, you can see that the mycelia of the fungi. So the mycelia is 2 groups. One is septated mycelia, and another group is aseptate mycelia. So these are 2 major groups. One fungal group belongs to that septate group, septate mycelial group. Another is aseptate mycelial group. And they are multiplying in terms of asexual and sexual means. So in sexual reproduction, they form some fruiting structure. So we'll be discussing in next slide how the fruiting structure looks like. And if you see that spore categories on very right hand, you can see that the spores are either single spore or many more spores altogether, and that spore should be single celled, multi-celled, and the size should be varied. Okay, so the next category is bacteria. If you see that plant pathogenic bacteria are usually rod shaped and that should be flagellated. So there is another separate lecture for the plant bacterial group. And the next group is mollicutes. I will not be going details of the mollicutes. So these mollicutes relating to the bacterial group, earlier it was considered as very closely related to the bacteria, and now they are separating mollicutes from the bacteria. The only difference, if you see that bacteria you can culture in the culture media, the mollicutes you cannot culture. And the morphology, if you compare, then the bacteria you can see there is a layer around the cell, but in the mollicutes there is no layer. And you can see there are some flagella in the bacteria, and in the mollicutes there is no flagella. And next group is plant parasitic higher plants. So plant parasites are also considered as plant pathogen. So I'll give you very details in next couple of slides. And the virus, they are very ultra microscopic. And if you see further, there is a viroid group. So the difference between viroid and the virus is only the coat. There is a coat protein that remain in the virus, and in the viroid there is no coat protein. And the last group is nematode. See, the nematodes are different shape and size, and they got eggs. They germinate from the eggs, and there is a juvenile group and an adult group. 3 stages of that development of nematodes. And protozoa actually is not our area of interest. So that fungal disease, the fungi as a whole, it belongs to the kingdom fungi, and as I mentioned that they are multiplying in terms of sexual and asexual means. On top of the picture, if you see that this is a sexual fruiting structure. In that sexual fruiting structure, you can see that there are some asci, the long threads, they are called asci. In that asci, there are 8 spores developed. That spores are called ascospores. Okay, so on top they are fruiting structure, sexual fruiting structure, and in the down they are asexual fruiting structure. And that asexual fruiting structure, they produce conidia. So on top they are spore and they are conidia. And why this fungal group are very catastrophic? Because of their proliferation. If you see that a single tiny fruiting structure, they produce enormous amounts of spore. So only single spore is needed to cause a big loss. Okay? So their proliferation is very important for considering this fungi as a big catastrophe. And the latent period, so you know that once the pathogen attach with the host, then it takes time to express their symptom. Maybe it should be 1 week or 1 day or maybe month, we don't know. So that starting from the attachment and penetration of the fungus, and at the end is symptom expression. So in between it's called latent period. So variation of the latent period is important to appear it as a serious disease or less likely. And there are various categories of spores. We know that there are some spores are airborne, some spores are waterborne, and there are varying category of the spores, which are very important for considering this as a catastrophic pathogen. And they are secreting some toxins and in some cases some growth regulators like cytokinin and gibberellin in some cases. So they are also combining together, considering this as a catastrophic pathogen. So this group separating from the fungal group is called oomycete or water mold fungal groups. So you all know about the late blight of potato. Earlier late blight fungus, Phytophthora infestans, considered as a fungus. But now if you see that it's now separate group, it's called oomycete group, and the kingdom is Straminopila. The fungal group, the kingdom is fungi, but this is Straminopila. Why it's separating from the fungi? If you see that fungal cell wall composed of chitin, but cell wall of this group, it's composed of cellulose or beta glucans. Okay, and in that sexual reproduction, fungus, they formed ascocarp or the fruiting structure, but here they produce oospore. If you see that it produces oospore. And down, this is the sexual, asexual means of reproduction with the zoospore. Okay, that zoospore and fungal conidia, they are in different group. Okay, so these are some differences between the oomycete and the fungi. And plant bacteria, I told you that most of the plant bacteria, they are rod-shaped and single-celled, and you can see that most of the plant bacteria are flagellated. There are a lot of flagella, maybe unipolar or multipolar, and only one group is separate, the Streptomyces down, is separating structure than other plant bacterial group. And the symptoms that produce, mostly they are gall-like symptom. You know that Agrobacterium, bacteria named Agrobacterium, it produces that crown gall. And some wilt symptom also produced by that bacteria. And some leaf spot and some scab symptom in the potato, they are all produced by that bacteria. And you know that it has different kind of, if you grow in media, it has different kind of colony they form, okay? And in the plant parasitic nematode, it belongs to Animalia group. It's called animal, okay? So the plant parasitic nematode, the major characters is its stylet. If you see that red circle, is the stylet. So this stylet separating these from the animal nematodes. In the animals, even in our human body, there are so many nematodes, okay? But if you separate it from the plant parasitic nematode, then definitely you should find that stylet. And out of 17 orders of nematode, only 2 orders belong to that plant parasitic nematode groups. These are Tylenchida and Dorilaimida. And in total, it was causing around $9 billion US per year. And another important character is that there is no segmentation or no septation, and the body is separating to the both ends, okay? So these are major characters, and one of the major symptoms is root knot nematode, root knot gall symptom. Why? Because this nematode secretes some growth promoting substance. That is why the cell development of the roots increased and it produced this kind of symptom. And the resultant effect is that the plant growth will be hampered. Plant virus, actually you know that virus is nothing but the genetic makeup, DNA or RNA. But in the plant virus group, with their DNA structure, RNA or DNA, it also has some coat protein. Okay, that coat protein is called capsid. So that TMV, tobacco mosaic virus, this one, it was invented long before. Maybe this is the first invented plant bacteria. And if you see the ultramicroscopic structure, you can see either that should be spherical or that round shaped. Okay, and this plant bacteria causing some common disease symptom. If you see that mosaic pattern of symptom development in the leaf, and the leaves are not that oil even, that should be some crinkle or stunted growth of the plant. So these are commonly symptoms that appear due to that plant virus. And there are separate groups of insects which are actually acting as vectors. What vector? They are carrying plant virus. So plant virus is not itself disseminating, it requires some vectors. So one of the major vectors is insects. And another group is nematode, and even human also carrying plant bacteria with their operations. But major group is insect group. And that insect mostly, see the red color, is Hemiptera. Hemiptera group of insects are the major plant vectors. And if you see the image on top, these are aphids. Then the whitish are whitefly and some hopper. And also at the bottom, these are thrips. So all of these group of insects, they are acting as a vector. They are carrying plant virus from one plant to another plant. How we transmit that virus? If you take a look at this picture, you can see that three classes of vectors. One is non-circulative, another is circulative but non-propagative, and the last group is circulative but propagative. In the non-circulative, the bacteria remain only within the salivary gland, not it pass through the hindgut and midgut of the insect. So once they take bacteria from one infected plant, and if they infect to another plant, then they are carrying the virus. And the non-propagative group, they are not multiplying within the insect, but in the propagative, the bacteria itself, they are multiplying within the insect, in their hindgut or midgut, or in their hemocoel. So plant parasitic higher plants, they are also considered as pathogen. I will be giving a very good example of plant parasitic higher plant in next half an hour. So if you just take a look, then you can see that there are two categories of plant parasite. One is stem parasite, another is root parasite. So did you see any of this parasite before? Anybody? Okay, yeah. So we will be giving a very good example of that plant parasite. There are two other categories. One is facultative and another is obligate parasite. So in that obligate parasite, if this parasite completely depend on another plant, it's called obligate parasite. And in the facultative, partially they are depending on the host. If you see that at the bottom, it has that green chlorophylls so that they could produce their food, but there are other nutrients maybe they are taking from that plant. So they are facultative parasite. So plant disease, we know that there are some disease, but do you know if plant feel that they are sick or not? Is it possible to understand? Any idea? So this is one of the major things that plant cannot understand if they are sick or they are not. Okay, it's not possible. But we know that if plant remain under stress, then they will release some ROS thing. ROS means reactive oxygen substance. So it's an advanced stage. We should not discuss about this. But plant disease, once it occur into the plant, it's not possible to understand if it suffered with the disease or not. We can only understand once we see the symptom, and sometimes that symptom's appearance would be a catastrophe. At the very last end, maybe we could figure out that the plant has suffered with the disease. Okay, and besides of its qualitative loss, it also secretes some mycotoxins that is really harmful for the plant or animal intake. Okay, so they are called mycotoxins. So we'll be discussing also about the mycotoxin. And disease triangle, this is a very good idea to understand about plant disease. There is a scientist, his name is George McNew. Since 1960, he thought that a disease should require a host, pathogen, and environment, but that host should be susceptible. If the host is resistant, then there will be no disease. And pathogen, that should be virulent. If pathogen is non-virulent, then there will be no disease. And the third component is environment. Imagine if environment is not suitable, then it's not possible for the pathogen and host to occur that disease. Okay, and for disease development in an epidemic manner, definitely all these three components should be working together. And the severity of a disease depends on that five categories. One, just we discussed about the environment. See the severity of the environment, because the host and pathogen, their development requires favorable environment, mostly suitable temperature, humidity, or rainfall sometimes accelerate the infection process. And the duration of the infection period, we discussed about the latent period. So if the pathogen needs only few days to exploit it and to develop, then there will be a catastrophe. And if it requires a long period of time, then definitely there should be less disease. And the prevalence of the pathogen, if it's appear in an enormous way, then more disease. And virulence of the pathogen, we discussed that aggressiveness of the pathogen is very vital for development of this disease in a vast area. And age and maturity of the host plant, you know that the earlier age or the seedling stage or the very tender age is very important, so that pathogen can easily penetrate into the host. And if it is root-borne disease, then roots are important. And if it is in above ground part, so if it is in late maturity, then pathogen will be less likely to infect. Okay, and the stage of the plant is also important. There are some pathogen attacks into the leaf, some pathogen attacks into the flower. Okay, so it's it's varied with the age and maturity of the host. And there are some hosts inherently they are susceptible to the pathogen. Okay, maybe their absence of the resistant gene. Okay, so all these components are relating to the development of disease in an epidemic way. And we should know about the cycle of the disease. It's also called infection cycle. If you understand about the cycle, then you can understand about the disease, how it occurs. It's not only a single step. So it starts from the attachment of what? Conidia or spore, right? So if you see on your right side, there are two groups of pathogen. One is non-pathogenic, another is pathogenic. So in the non-pathogenic group, they are not, although pathogen is attached or conidia is attached, but it could not penetrate. Okay, so the host should give some response. So after that attachment, host should recognize the pathogen. If host is not recognized, then there will be no disease. So attachment followed by host recognition, then germination. After recognition, pathogen will germinate and enter into the host. So penetration followed by infection. Infection means that establishment of the pathogen into the host. And once it established, then it produces the lesion. See the symptom of the leaf? Then once it produces symptom, then there will be matter of sporulation, means its reproduction. I told you that fungus reproduce through sexually or asexually, making some, see this are the fruiting structure, and only a single fruiting structure contains thousands and millions of spores. And the symptoms of the disease, if you just take a look of this plant, on your left side it's all healthy conditions, okay, and the metabolic process, photosynthesis, everything going well perfectly in the left side. But in your right side, it's all disease condition. Starting from the root rod, if the roots are disturbed, then plant's growth will be disturbed. Then if you see that crown gall symptom, if the crown gall symptom appears, then there will be no exchange of the nutrients and water. We know that water comes from the soil to the leaf, and after that photosynthesis, nutrients will come from the leaf to the down. So if crown gall appears, then there will be disturbance of transmission of food and water. Okay. Then you see that vascular wilt. Vascular means the pathogen will affecting the vascular tissues, like xylem and phloem tissues are infected. Then there are some symptoms also like leaf spot or canker or fruit rot, or altogether it's called leaf blight. If multiple spots are accumulating together, it's also called leaf blight. Okay, so all the symptoms are on your right. If you compare it with the left, you can understand the disturbance of the plant growth. So plant disease epidemic. What is epidemic? It's just sudden outbreak of the disease. So if the disease appear in a large scale, in a large number of plants, then we can tell it as an epidemic. So in that issue, we should also know two other terms. So one is endemic, another is pandemic. So if the epidemic appears in a particular region, okay, then it's called endemic, means it's appear in a small scale. And if the disease appear in a large scale, means it covers multiple continents or multiple countries, then it's called pandemic. Okay, so this epidemic either would be endemic or would be pandemic. So to understand about the development of epidemic condition, you should know about the graph on top. You see that there are two cyclic conditions. One is monocyclic, another is polycyclic. So in monocyclic, there will be only one cycle of pathogen development in a growing season. But in the polycyclic, there will be multiple cycle in a growing season. But all if you see that the static condition after development of the exponential curve, so from the lag phase, then exponential phase, and then static phase. So why the disease becomes static at a certain period? Anybody can tell? Any idea? Because once the pathogen infect the host, then maybe it takes all the nourishment. Once all the nutrients from the plant finished, then definitely that should be in static condition, right? Means that it's completely damaged. So there will be no point of development of the disease, right? So that should be then static. So we discussed three components of the disease: host, pathogen, and environment. And there would be another component, is time component, because in a season it's varied with the disease appearance. Okay, it depends on either it's windy or it's rainy or humid. So the time, you should also consider the time to consider this epidemic disease development. And in this book, Plant Pathology, Agrios GN, he also mentioned about fifth factor. So this is also a very vital factor. We should not ignore contribution of the human. Okay, so I will ask you, any of you can tell any idea about the human contribution to the disease development? Yeah.
Do you mean things like what sort of pesticides and chemicals a farmer might use on their
Yeah, that should also be counted. Yeah. And I will be adding here that if you, human being, carrying say infected seed from one place to another place, okay, then you are the fifth factor, right? And see that two images, that operation of the plant, because you are using some tools. If these tools are contaminated, say for instance if it is bacterial disease or viral disease, say you are operating some of the agricultural practices, then you are risking other healthy plant to be contaminated with the pathogen. Okay, so definitely human contribution would be a vital for development of the disease. And as she also mentioned about the pesticide, so you should decide either you will be applying pesticide or not. If you don't apply pesticide, then plant will be suffered a lot. Okay, so we should not ignore the contribution of the human being. And the climate change issue is also an important thing for the plant disease development. So we know that during the climate change there will be a massive accumulation of the carbon dioxide and methane gas and a lot of things. So carbon dioxide is very important for what? For photosynthesis, right? So for photosynthesis, carbon dioxide is vital. So if carbon dioxide is released, then there will be possibility of more photosynthesis, more plant development, and if plant is developed a lot, then there will be possibility of infestation by the pest and pathogen. Besides that, environmental catastrophe like raising of the sea level and some other issues, these are also some partial issues of infestation by pest and pathogen. So if you just write, I'll show you some slides. You just take note, then I will be asking which is what. So what is this? This? Bacteria. This? Fungi. Good idea. Any idea? Is it fungi? Bacteria? Nematode? Parasite? Parasite? Yeah, so this is plant parasite, okay. What is this? Fungi? How many of you fungi group? Okay, Lord, yeah, you're right. So same thing with the mushroom, also in fungi group, and this root rotting fungi, okay. Parasite, insect, but what group? It's vector, right? Insect, vector. Okay, it's insect, but it's vector. These are thrips. Okay, but with that image, it's not possible to understand. But these are insect vectors. It's carrying bacteria from one plant to another plant. What are these? Weed, yeah, right. Okay, so these are weed, and you can consider it as a parasite. And there is a break for 5 minutes. Okay, then we'll be discussing next part.
And I changed my topic to that. So after that, there are many countries involved in that. And it's Mexico, India. Yeah. It's from, but that actor from United States, and they do that in Mexico, right? Is that correct? And it's like it's original Korean Revolution. It means the first time, not the second or the third one, right? I think it's just the.
It started from 1930 towards 1960.
Only for
In between before Second World War to after Second World War.
So we only talk about the
Period of Green Revolution. But now it's continuing with the Green Revolution, but the beginning in between 1930 to 1960, in one article I've seen that it's from 1930 to 1960.
only that period, right?
That period is revolutionary, okay? But that revolution is still going on because still we are using high yielding variety, pesticides, and a lot of things. But the beginning point, that time, and there was massive revolution, and they could see that how crop is produced. Okay, and a lot of crops are produced, and they could minimize so many things. And there was invention of DDT, you heard about DDT? So DDT and some other fungicide and pesticide, it's all actually started from that part.
I also searched about at the Korean Revolution, and here's on like the second. Do we need to mention that?
Second what?
Second Korean Revolution.
Oh, second Green Revolution. Yeah, if you could relate, then you can tell.
But the instructions that we talk about origin Korean Revolution.
Okay, you can relate. It's possible you can relate.
Like
What the Phil Salisbury mentioned in last lecture. Did he go through second Green Revolution or you didn't follow his lecture?
okay.
Yeah, you can go through and check at what point we was discussing.
So, like from the first Korean Revolution, we get some like effect of that, like the environment or some the erosion of the salt. So after that, we changed the plan or something like that for the next green revolution. So like that. So the introduction says the outline the green revolution. Can I just say like the green revolution in different countries like Mexico, and I can list the
Did you discuss with Andrea? Andrea, did you discuss about this issue?
No.
Okay, so I can start the second part. I think by this time you knew a lot of things about plant pathology, right? I don't know whether you could capture or not. But if you understand the basic of the plant pathology, now it's all about the application of the plant pathology, starting with the diseases that appeared in an epidemic way in many countries, and it caused a lot of sufferings for the food safety and for human civilization. So this is a review article. If you get time, then go through with this article. It mentions about some emerging diseases that appeared in last 2, 3 or 4 decades, and it's mentioning all about the developing countries. And we'll be discussing many of these emerging diseases in next few slides. But I will be mentioning about some key points why the emerging diseases are most prevalent in the developing countries. Anybody get any idea about that issues? The developed countries versus the developing countries. Any idea? Yeah?
Is there more humidity? More rainfall?
Not that much. And another one you said on your right? Anybody? No. Okay. So if you compare with the institutional things, that the developed countries, they are more stable. Their infrastructures, their planning, and their policy is very precise and very nicely done, right? In compared to the developing countries, there's no institutional structures, and the agriculture policy and some other policies, it's fragile. Okay, it's not that well done. And in the developing countries, there was a tendency to migrate people from the rural areas to the cities. And their belief that if they could migrate from the rural areas to the cities, then there would be a better life. But in other sense, they are risking the agriculture operations and agricultural activities in danger. Okay, we'll give a short description in the next slide why it happens. And there was a price hike of the seed and other agricultural inputs, like raising the price of the fertilizer and pesticide. We all know that the pesticides and fertilizers and some good quality seeds, they come from the developed countries, and the developing countries, they depend on that agricultural inputs from their developed countries. So if the price hike happens, then definitely there will be some risking of that disease development. So plant parasite, I think now you can get better understanding what is plant parasite. But I will be mentioning the severity of that striga in sub-Saharan African countries, because as I just mentioned that the migration of the rural community from the rural areas to the cities, so people risking the agricultural operations in that rural areas. That's why earlier they were practicing that weeding and some other agricultural practices, but they left their land. By this time, this plant parasite developed enormously, and now this stage is completely, in some areas, is completely unable to protect or unable to control. Why it's severe? Because this striga, they are root parasite. So they take all the nourishment from the plant roots. So it's not possible to develop the plant, right? And they are also secreting some poisonous substance. So that's why the plants become poisonous. And another important thing is that the seeds of this plant parasite, they could survive a long period of time, so that if you practice crop rotation, then you cannot prevent this infestation. Okay? And that's why it's called witch's weed, and it causes around 50 million hectare of lands, and the loss amount around $300 million. And Nigeria, in that sub-Saharan African countries, is the worst affected. And interestingly, in Australia, there are some biosecurity issue, mainly in Queensland. They are declaring that this is a very threat for the agriculture in next couple of years. That's why they are asking it to protect, and we'll be going through a video clip to understand about this striga.
Red witchweed, also known as Striga asiatica, is a parasitic plant that could pose a risk to Queensland's agricultural industries. Hosts of red witchweed include economically important grasses and summer cereals such as sorghum, corn or maize, rice and sugarcane. Unlike most weeds that compete with crops, red witchweed damages plants more directly by tapping into the host plant's root system and taking its nutrients and water. The host plant then wastes energy supporting red witchweed growth rather than its own. Red witchweed grows between 10 and 40 centimetres tall. Its leaves are arranged in opposite pairs along the stem. Leaves are 6 to 40 millimetres long and 1 to 4 millimetres wide, and have a tapered, pointed tip. Its flowers are 5 to 20 millimetres in diameter and are usually red. However, white, yellow or pink flowers have been found overseas. Red witchweed reproduces from seeds. The seeds are tiny and dust-like and can easily drop into the soil, enabling its spread. A single plant can produce between 25,000 and 200,000 seeds. The minute seeds are mainly dispersed by wind, water, soil movement, or human activities. When a vehicle or machine is driven through a weed-infested area, seeds can become lodged and carried considerable distances before dislodging in new areas. Red witchweed can survive and produce seeds in a wide range of soils, but generally prefers free-draining, sandy or gravelly soils. The most likely place to find red witchweed in a commercial crop is in these soil types. You should observe a property hygiene protocol and avoid moving equipment and other things that might carry soil if not appropriately washed down. Adequate wash down of machinery and equipment and other biosecurity precautions can reduce the risk of spreading red witchweed. Red witchweed is a notifiable pest plant in Queensland. If you find it on your property, you must report it to Biosecurity Queensland immediately on 13 25 23. Remember, early detection and...
Okay, thank you. Yeah, so now you can understand about the infestation of witch's weed. So you can get other video links about the sub-Saharan African striga infestation in YouTube. So now I will be discussing about the bacterial wilt of banana. So in Ethiopia, 40 years ago, this bacterial infestation was first reported, and about 2 to 4 billion US dollar loss due to that disease. Why? Because once it appears, then you can see that there was premature development of the fruits. Fruits are not matured, and wilting of the whole plant. If the plant become wilted, then there is no yield, right? And the symptoms, if you take a look, then the placenta of the fruit, see that black to brown discoloration, and in the pseudostem you can see that bacterial ooze, they are coming out from the pseudostem. So besides this bacterial wilt, I was also mentioning about the Fusarium wilt, it's fungal wilt. So if you compare with the fungal wilt and the bacterial wilt, you can see the difference. So in that pseudostem, the vascular tissues, they are filled with that bacterial ooze, and in the fungal stem, it's the vascular tissues, they are actually rotting or brown to black discoloration, and both of these wilting cause enormous crop loss across the world. But most importantly, in the sub-Saharan African countries, Xanthomonas bacterial wilt is now presently it's very severe disease. So brown spot of rice is a fungal disease. The fungus name is Cochleobolus meardibinus, and see the spore. These are the spores. Yes.
I just have a quick question about the bananas. Is that the same disease that affected a farm in Far North Queensland? RF something 2F5?
I'm not sure about that. So are you sure that these are bacterial wilt?
Yeah. Well, no, because it says Panama.
Oh, Panama disease. Yeah.
Saying that we had here.
Maybe because that Panama wilt disease is also fungal disease. So that fungal wilt is called Panama disease. So this brown spot of rice, there was one of the biggest famine that happened in Bangladesh, India, and that subcontinent. So that's why it's called Bengal famine. it caused 2 million people died during that period. Okay, so only one single disease can cause a huge loss, huge sufferings for the human civilization. So this was one of the biggest threat at that time, and this is caused by a fungal pathogen. Okay, you can see sometimes the name of the fungus. It was earlier Bipolaris. Now it's changed. The naming has changed to Cochleobolus. Okay, in earlier literature, if you see, then don't confuse, because the taxonomy and the naming has changed. Okay, so now this is the latest name of this fungus, and this was also causing damage to the leaf. If a leaf is damaged, then definitely it will be causing damage to the grains. If you see that grain quality, the left side healthy grain and the right side they are infected grain, and in this grain they are carrying the fungal pathogen. So if you transplant this seed to the next season, then also fungus carrying to the next season. Okay. And another epidemic disease development nowadays in Bangladesh, India subcontinent, this is called wheat blast. If you just take a look of these two pictures, these are very latest, maybe within 1 or 2 months time. So it was just appeared in 2016, appeared in Bangladesh. Then in 2017, within 5-7 months time, it migrated to the India. So in India there is another Bengal area, it's called West Bengal, and they were predicting that this disease could migrate to Indo-Gangetic plain, the whole subcontinent, including that Myanmar, Vietnam, all rice-growing areas may be infested by this disease. So why it's very severe? Because once it appears, then see the infertile grains. So the grain loss completely it appears. If this disease, then there was a report that 100% crop loss occurred. Okay, and farmers were very actually frustrated, and they were burning their crops. And this is one of the measures to protect the disease from one season to another season. So in which situation they could burn their own crops. Okay. So see the devastation and the severity of a particular disease, right? So interestingly, this disease first reported in Brazil since 1985, but it transmitted to another 2-3 subcontinent, right? So the source of transmission nobody knows, possibly through the seed or through the wind. Okay, it's not clear how it migrated, but see the devastation from one continent to another continent. Then blast of rice. So earlier it was blast of wheat. Now it's blast of rice. It was appeared in an epidemic manner in Italy, and majority of the European countries, they were experienced a lot with this disease. And if you just take a look at the maps, you can see that most of the African continent and the Latin Americas and the Europe and part of the Asian countries, they were suffering a lot with this disease, but comparatively free in United States, and definitely Australia is one of the safest to occur this disease. And wheat disease, UG99, I think many of you already heard about that, UG99. Anybody get any idea what it is, UG99? Any idea? So, okay, UG means Uganda. So, this disease, actually it was appeared long before, but in Uganda, since 1999, it was appeared in an epidemic way. Why? Because SR31, see on top, SR31 was considered as a resistant gene for this pathogen, but this pathogen, they were breaking that resistance gene. Okay, so this resistancy was no longer working in Ugandan isolate. That's why it was appeared a huge amount of loss in that Uganda. Okay, and if you just take a look at the maps down, you see that in the middle is Uganda, and see the migration of the disease from one part to the another part of the African continent, and it was migrated to the Mediterranean region and also migrated to the Asian subcontinent in that 10-15 years time. How is possible to migrate? Anybody can tell how the fungus can migrate from one continent to another continent? Human, yeah, definitely. And it's airborne. If it is airborne, then through the air it could migrate from one continent to another continent. And with the human being, if they carrying infected seed from one place to another place, in that way it also could migrate. And in that rust condition, there are two cyclic conditions. One is macrocyclic, another is microcyclic. As I mentioned here, that see that rust completed its life cycle in 5 stages. Okay, so that 5 stages are Basidiospore, Pycnospore, Achiospore, Iridospore, and the Teleospore. This is the whole cycle. So if it is completing with the 5 cycles, then it's called macrocyclic. Okay, and what is the microcyclic? In that microcyclic, not that 5 stages, maybe 2 or 3 stages, possibly 2 stages, Iridospore and Teleospore. Okay, so these are difference between macrocyclic and microcyclic. Okay, and for the host, particularly in this wheat rust, it needs 2 hosts. One is barberry and another is wheat. So that's why it's called heteroecious. Okay, and there are some rust, not this rust, some other rust. It finished its life cycle using only one host. It's called autoecious. If you just take a look at the leaf in Basidiospore and Achiospore, they use that barberry leaf. So part of their life cycle completed within one host. Then from that host, it migrate their spore to original host, that wheat host. Okay, so one part finished in barberry plant, another part is completed in wheat plant. Okay, that's why it's called heteroecious rust. Okay, so if there is an issue of rust, then you will find these terminologies. Okay. So late blight of potato, as I said you that earlier it was considered as a fungal pathogen, but now they are separating it as a different categories. It's called oomycete group. So the causal organism is Phytophthora infestans. And you all know that there was the biggest famine in the human history caused by a single particular disease, and it was called Irish famine. Okay, so in Ireland there was also a monument, and there was an article, you all could find it easily. So, and another important thing is that there was the biggest migration in the human history occurred only due to that particular disease. About 1.5 million people migrated from Ireland area to United States and some other countries. So why this is a very important disease for appearing it as a catastrophe? Because it was multiplying a lot under favorable conditions. What's the favorable conditions? See that cool weather and humid weather. And if it is rainy conditions, then it could migrate, it could multiply easily and it could migrate easily. So and this pathogen could transmit through the water. Okay, so water, wind, and all sorts of things they could use, and they could develop symptoms very rapidly. And interestingly, they could carry that fungus through their tuber. Okay, you know, potato, they actually multiply and develop through the tubers. So if the tubers are infected, then in the next season they will carry the infection. And if you just take a look there, it was started from 1845, and that disease was continuing for 4 to 5 years. Okay, and that Irish people, they were solely depend on this potato crop. They were not thinking about any other crops. Okay, so they were every year they were cultivating only a single crop, and they were collecting the seeds and the potato tubers, and their food and other things completely depend on that particular crop. So once that appear in an epidemic way, then they thought that in the next season maybe it will not appear. But as the potato tubers infected, then it's also carrying their infestation to the next season. Okay. Likewise, it was going 4 to 5 years time. At that time, there was no charity and no other thing to feed them, right? So that's why it was causing enormous loss. Again, I will be showing the last video for today to give you an understanding about the life cycle of the fungus. Sorry, where it is.
Could be latently infected by the late blight pathogen. Between starch cells inside the infected seed tubers, hyphae of Phytophthora infestans grow, while on the outside, the tubers look completely healthy. As soon as the infected tuber is latently infected by the a Phytophthora epidemic often originates when potatoes are being planted. Even if certified seed is used, it's still possible that individual tubers could be latently infected by the late blight pathogen. Between starch cells inside the infected seed tubers, hyphae of Phytophthora infestans grow, while on the outside, the tubers look completely healthy. As soon as the infected tuber begins to germinate, the Phytophthora mycelium grows into the sprouting shoots. If the degree of infection is not too high, the plants emerge and survive without any sign of bearing the pathogen inside. It's not until a few weeks later that the first symptoms become visible. Brown lesions, especially at the base of the stem, but also in the leaf axils, where overnight dew formation encourages growth of the pathogen. Soon afterward, small brown lesions form on the leaves. They often have chlorotic borders. In humid weather, these necroses, found especially on the edges of the leaves, expand rapidly. On the underside of the leaf, whitish growth of the pathogen can typically be seen. It consists of a dense aggregation of branched hyphae, sporangiophores, bearing lemon-shaped structures on their tips. These sporangia are the asexually formed reproductive organs of Phytophthora infestans. A few infected potato plants are sufficient to cause an epidemic. Continuously humid weather favors the spread of Phytophthora infestans.
Thank you. Okay, thank you. I think you understand now about the potato late blight. So now I'll be going through that southern corn leaf blight. So why it's southern? Because this disease was appeared in the southern territory of the United States, and this is also a fungal disease, and with that infestation it could completely lose the whole product. But we are lucky that at this stage it's not possible to appear cause any famine, because it's United States and there are a lot of charity organizations nowadays, and it's not possible to appear as a famine. But anyway, it caused a huge loss. And the cassava mosaic disease, this is a viral disease, and we know that the plant virus are either DNA virus or RNA virus, but this is a single strand DNA virus, and the vector of this virus is whitefly, and it was appeared as a famine in Africa since 1920. And if you take a look at the symptom, that it was actually the flesh of that cassava, it was that brown necrotic lumps it formed, and it was secreting some toxic substance. That's why it's not possible to feed and cause that devastation. So until this one, I was covering 9 different emerging diseases, but there are many more, but I was trying to highlight you some important diseases around the world. So now I'll be discussing about why these diseases were appeared in an epidemic manner. I was discussing a little bit about the cultivation of the single cropping. If they are completely depend on a single crop, then every time there is a possibility that disease could migrate from that potato infected tuber or from the infected seed to the next season. So if you completely relying on a single crop, then there will be possibility of appearing this as a famine. And favorable environmental conditions are also important factor in cultivating that infected seed and tuber to the following season without any treatment. And if you don't understand the disease cycle and don't understand the disease triangle, then you will be also risking your plant. And if you don't use chemical control, if it is emergency, then you should use chemical control, chemical pesticides to protect your crop. And in some cases, the disease will appear in a mass. Okay, so at that stage, it's not possible to control the disease. So the last few slides were all about the pre-harvest pathology, pre-harvest disease. Now I'll be focusing on the post-harvest pathology and post-side.
I'm just curious if you can speak to the emergence of superweeds.
Superweeds?
Yeah.
Yeah, as I told you that it's not possible to cover everything. So weeds should also be an important factor. But my focus was mostly on the plant disease, and I was covering a little bit about the plant parasite. So yeah, you are right that there are some weeds you can think of. Yeah. So for the post-harvest pathology, the disease that caused by the post-harvest pathology mainly through the contamination of your products with using that contaminated tools, and once you harvest, then if you make any injury and if you don't pack your product properly, and if you don't maintain that hygienic conditions, and if you don't maintain your storage conditions safe, then there will be possibility of more infestation by that post-harvest pathogens. And if you just take a look at the list of the post-harvest organisms, it's include fungi, oomycota, oomycetes, and bacteria. Okay, so all these, along with that insect pest. Okay, I'm not focusing on the insect, but insects are also contributing here. But as my target was mainly to focus on the plant disease, that's why I am covering these plant pathogens. And besides that quantitative loss, this post-harvest pathogen also contribute to the quality of the products. We know that mycotoxins, I will be discussing more detail of that mycotoxin, but that mycotoxin actually appear mostly at this stage. And for the developing countries, they are suffering a lot compared to the developed countries. Okay, and these are a very brief idea about the post-harvest decay of the fruits. Only these are not the post-harvest disease. There are many more and many more hosts and many more symptoms. But in brief, just to give you a very glimpse of the post-harvest fruit loss, post-harvest disease of fruits. Did you see any of this symptom in your fruits before? Anybody get an idea about these symptoms? I think many of you, right? So these are all fungal pathogens. Besides that, there are some bacterial pathogens also causing that fruit loss. And in the vegetables, I think some of you can get idea about the mold disease, you know. So there are different names of the mold: blue mold, black mold, gray mold, green mold. So that blue and green mold caused mostly by the penicillium, and black mold mostly caused by aspergillus, and gray mold by the botrytis. Botrytis fungi is this one. So these are all common mold across the world in that post-harvest vegetables. And bacterial soft rot, this is the most important post-harvest bacteria around the world. Okay, so its naming is Pectobacterium keratovorum. Earlier it was named as Arowina. So why Pectobacterium? Because once this bacteria attacks, it degrades the pectin material of the cell wall. Okay, and so that you can easily see that cell collapse and degradation, and popularly is named as soft rot of potato, or soft rot of onion, or soft rot of cabbage, cauliflower. Okay, so how can you identify this bacterial symptom? If you just smell it, then you could experience bad smell. Okay, and that rotting symptom should be that should be dry, it's sort of wet conditions. Okay, and woody plant tissues are not infected by this pathogen, and the conditions should be warm and humid conditions. If prevails, then this disease will appear enormously. So in the grains and legumes, mostly it's infested by that aspergillus and penicillium, like that mold fungus. And these grains, if it's infested like this, then they will also carrying some toxin. It's called mycotoxin. Okay, so if we now discuss about the mycotoxin and mycotoxicosis. So what is mycotoxin? Myco means fungus, toxin means the toxin. So if toxin released from that fungus, it's called mycotoxin. And with that toxin, the disease that appear due to that toxin is called mycotoxicosis. Okay, so there are many more examples of mycotoxicosis I'll be discussing in next few slides. And what's the importance of this mycotoxin? Because of its, if it appear, then it will cause a lot of damage and a lot of impairment in our human body, and most severely it's carcinogenic. Okay, So, and it will be break down our immune system. So think of the severity of this mycotoxin. So ergotism is a mycotoxicosis, the disease that caused by that ergot. It's a fungal pathogen. It's Claviceps purpurea. If you just take a look at the structure of the ergots, it looks like it's similar to the grain, cereal grain, right? So long before, since 857 or 850, it was appeared enormously in that European countries, most importantly in the France, okay, and Italy and some other Western European countries. And at that time they were considered it as a holy fire. Holy means because they were considered it as a punishment from the god. Okay, so they could not figure out the reason behind that pain. Okay, see that mummified legs of the animals, and also same symptoms appear in the human body. So if you just go through and check the symptoms in the human body, I got some very, that pictures and image. I was not like to show it here. So it's very severe, and you could feel extreme pain due to that toxin. So there are two categories of symptom. One is convulsive, another is gangrenous. So gangrenous ergotism is very crucial. And for the ergot, it's actually nothing but the compact fungal mycelia. Okay, so with the favorable condition, it could germinate and it could produce that Si and ascospore. Okay, so once it mixed with the grains, then it actually deteriorates the quality of the food. And aflatoxin released by that Aspergillus flavus. Afla comes from Aspergillus flavus. So it caused problem with the poultry as well with the human being. Okay, and it easily actually contaminate the grains. And Fusarium head blight. So it also a fungal problem. See that Fusarium oxysporum or Fusarium graminearum. So once this fungus infect the grain, you can see that it's also causing the quantitative loss, right? So in the field, it causes quantitative loss of the crop, and also it risking the food for the quality. Why? See that three mycotoxins named trichothecin and that zearalenone and DON, like deoxynivalenol, okay, that DON popularly is named as DON. So all these mycotoxins actually deteriorating the food. So with that disease, it causes qualitative loss and quantitative loss. Okay, and what else? Toxins like ochratoxin and tremorgenic toxin and petulin is mostly released by that Penicillium, Aspergillus. Okay, and you could experience a lot of problems in the human body as well as in the animal kingdom. And Salmonella. So in recent time, scientists from the United States, they were collecting a lot of post-harvest fruits and vegetables, and they were culturing from that source, and they found that around 68 to 72 in some cases infestation by the Salmonella. And you all know about the severity of the Salmonella. If not, then in next few lectures, I think in the animal pathology, you could experience the severity of the Salmonella. So in my topic, I think that you should know about the possibility of infestation by that Salmonella. That's why I'm giving this slide. So to avoid that problem, you should take some pre-harvest control measures. Means that if you are not injured your product using that infected tools, and if you regularly apply pesticide, and if you make your product free from fungi in that pre-harvest condition, then there will be less likely to appear it as a disease in the post-harvest condition. Okay? And you should know about the process of diagnosis. Okay, we covered with the plant disease and all about that emerging disease, but how can you identify, how can you diagnose? We should know a little bit, right? And the reason for diagnosis mainly to identify the causal organism. If you know the causal organism, then you could take better control option, right? So because that fungus, bacteria and virus, the control measures should be different. Okay? And for the quarantine purpose, we should know the cause of the organism. Otherwise, how can you take the quarantine measures? So these are the purpose. And for the diagnosis, the traditional way of diagnosis is to see the symptom. I was providing here a small symptom is caused by the brown rot, and any of the pathologists with that symptom expression, they could figure out that this should be brown rot. But with that traditional way, it's not possible to figure out the symptom or the cause of the disease by any of the people, those don't have any idea about the plant pathology. So the traditional technique is very important for identifying the disease, but you need some skill. You need some background knowledge about the disease and pathology. And for the identification of the disease, it was Robert Koch who has invented or developed 4 pustules to figure out a particular organism is responsible for a particular disease, and he proposed 4 different steps to confirm that a particular pathogen is responsible for a particular disease. So if you follow through that 4 steps, then you could identify a causal organism. So at that time it was a big revolution for that disease diagnosis. So there are 3 questions. If you write it down, or I'll tell you after showing that slide. What is this? Number 1. Number 2. Number 3. Now you can differentiate. Which is what? What is number 3? Disease, right? Number 2? Nutrient, yes. And number 1, insect. So if you just compare with the 1 and 3 with the number 2, if it is nutrient deficient, then the whole field should be showing almost same symptom, right? Because one particular nutrient, if it lost, then it might be showing similar kind of symptom in the whole field. But if it is infested or infected by the whole pest or pathogen, it will show that symptoms, that not regular basis. And it's easy to identify insect infested field because you can see the insect around the infected area, infested area. But it's not that possible to determine plant pathogen, because sometimes you might not see any of the fungal structure around or bacterial structure around the infected area. So another very quick, number 2, number 3, 4, 5, 6. What is number 1? Easy, it's insect, right? Insect. And number 2 is a bit difficult. Virus, right? Who said virus? Why virus? Because it's sort of easy. You can see that the leaf size should be shorter, stunted growth, and some mosaic symptom. Okay, sometimes it's difficult to differentiate, but these are some symptoms. And number 3, bacteria. Yes, why? See that these are ooze, bacterial ooze. As I said before, that in Xanthomonas wilt disease in banana, you could see some oozing coming out, right? So in that bacterial symptom, sometimes, it not always, sometimes you can see that bacterial ooze coming out. Number 4. So these are fungus. If you take a look, then you can see that the fungal structure, that mycelia, that whitish mycelia, not always, sometimes you can experience. And number 5, easy. Insect, yes. Because you see that only insect could take the whole tissue out, right? It's, and for the 6, nematode, yes. Because previously I also showed that problem. So you see that these are root gall symptom, and it's commonly occurred by that nematode. So all these were part of that disease identification using that symptom. And there are some molecular approach. You all know about the PCR, and there are some multiplex PCR. So in the right of the image, you see that 2-3 organism, if identify in a single reaction, then it's called multiplex PCR. So using that PCR, you could precisely identify the target organism. And in the RT-PCR or real-time PCR, it's not only identify the pathogen, but you can also quantify the inoculum in an infected plant material. So after diagnosis, we should know about the management practice. That management practice begins with that cultural methods, because we should not ignore these practices, because if you consider that host eradication, so if any plant infected by the virus or bacteria, it's not appear in an epidemic way in the very first stage, right? So maybe 1 or 2 plants would be infected. So if you just eradicate very carefully that infected plant, and if you burn it, then you can, at least you can save some of the adjacent plants. Okay? So that's why host eradication is also an important practice. And for crop rotation, we know that there are so many soil-borne pathogens, and so many pathogens, they could survive in the field for a particular growing season. And in the next season, if you plant same host, then this pathogen from that soil, they will germinate and will cause this disease. But if you change the host, then that pathogen will not get favorable host. Then you can protect your plant from further infestation. Okay? And sanitation, if you are properly managing your field, if you are using that that weedicide, and if you, there are some pathogens which could survive in the weeds. So if you protect your plant from that weeds, and if you eradicate all the infected materials around the plant, then you can be better practice your crop. And the conditions you should make unfavorable for the pathogen. How? See, take a look at these bottom 2 pictures. In the left side, it was well-maintained products, and the right, see that poorly managed. Okay, so if you make a very good storehouse conditions, if you pack your products very nicely and precisely, then you can avoid that infestation. Okay, And there is a very good practice using that, that polythene traps. Okay, so that yellow polythene traps attract by the aphids mostly. So if you kill the aphids, then there's two benefits. One is that you are protecting the crop from the insect, as well as you are protecting your crop from the viral infestation. Okay, so two benefits. So these are some traditional cultural methods you can easily practice. And for the biological methods, they are very recently, they are actually that we all know that we should avoid using pesticide, right? So you should take these measures like biological methods, but you should understand about a couple of things. One is the mycoparasitism. If you see that myco means fungus, so if you kill one fungus using another fungus, it's called mycoparasitism. Okay, and in the antibiosis, if you see that bottom two pictures, you see that bacteria could kill a fungus. Okay, because in bacteria they secrete some antibiotic, which actually controlling that fungus. Okay, and also the competition. So all these combinations actually using at this stage in the biological control approaches, and using that suppressive soils also carrying that beneficial organisms, so you can use that suppressive soil to protect your infested field. And these are the last method of disease management, and I think in last lecture in Phil Salisbury you understand about the breeding method, so I will not go through. The chemical control, there are so many pesticides nowadays available to protect your crop from the field, but you should know which are the causal organism, either it's bacteria or fungi or else. And plant quarantine. So in the developed countries, they are very, very now developed in that quarantine system, which is very essential to protect many of the diseases from one continent to another continent. And you are lucky that Australia is the number one top-ranked plant biosecured country around the world. That's why in Australia they are experiencing very less disease compared to other countries. And integrated disease management, they are combining all the possible combinations from that traditional, that conventional, cultural, biological, chemical and breeding, everything if you practice all together is called integrated disease management. Now the future challenges, this is the last slide. So the future challenge number one is that infestation by the Salmonella or E. coli, because these are now recently reported, it was not assumed before. And three terminologies, I don't know you acquainted about these terminologies, is bioterrorism, biological warfare and agroterrorism. Anybody get any idea? Just in a quick.
Yeah, purposely infecting something with Lyme disease.
Okay. So the bioterrorism, I will just give only one example. I don't know you could experience about the, since 2001 or 2, with the envelope in United States, they are carrying anthrax disease from one place to another. Okay, so they were targeting human civilization. Okay, they were risking the human to infect by that anthrax disease. At that time, it was severe in United States. They were taking proper action so that it's not spread out. And the biological warfare, we all experienced about that biological warfare since World War II and in Iraq-Iran war. So they're using that biological weapon. And the agroterrorism relating to that bioterrorism, say if you are targeting one continent, that you will be making that your neighboring countries to be infected by a particular disease, you know that the severities. Say you are carrying 1 kilogram of that infected seed, and by any means you could migrate it to your neighboring countries in an intentional way so that you can risking the agriculture in that particular neighboring countries. So in that case you can make that agro-terrorism. So in future we don't know what happens, and we should all aware about